Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatreen Sawati. And Shatreen Sawati is here to deliver the lecture about the gene library and its type. If you're not familiar with the gene library, DNA library, must watch my lecture. Do subscribe my channel, like my videos, and share my videos. Now, what is basically the gene library? Actually, gene libraries, like the conventional libraries, are used to collect and store information as a set of DNA molecule. Genomic libraries are commonly used for the sequencing applications and very important role play a very important role in what in whole genome sequencing, like the human genome and many other organisms have been sequenced. So gene library play a very important role. Now in this lecture, I'm going to discuss about the types of the gene libraries like cDNA library, genomic library, randomized mutant libraries, their differences, and also the protocol for the construction of these libraries. Okay, so first of all, what is gene library and how can you define the gene library? A random collection of the DNA fragments typically presenting the entire genome of an organism. Must keep it in your mind. The entire genome that have been inserted into the cloning vector. Gene libraries are sometimes called the DNA libraries or the gene bank too. Okay. Now the types of the DNA libraries. There are three different types. Number one, cDNA libraries, and cDNA libraries are constructed by using the reverse transcription mechanism. Genomic libraries formed from the genomic DNA, and the randomized mutant libraries formed by de novo gene synthesis, where alternated nucleotides or codons are incorporated. So I will discuss each and every thing, each and every library in detail one by one. So first of all, I'm going to discuss about the genomic library. How can you define the genomic library? A genomic library is a collection of the total genomic DNA from a single organism, means the full length genome of an organism. The DNA is stored in a population of the identical vectors, such containing each containing a different insert of the DNA, using a host cell like bacteria to carry the vectors, allow for easy amplification and retrieval of the specific clones from the library for further analysis whenever you need. Is it clear? Okay. Now, how is genomic library is constructed? In order to construct a genomic library, we need the DNA, which we can extract from the desired tissues or cell of an organism. Then we need the restriction enzymes for the digestion of that DNA. You can see here, these are the tissues cells from which the DNA is extracted, and we need the cloning vectors. And what will happen, these cloning vector along with this DNA is digested with the restriction enzyme. After cutting with the restriction enzyme, what will happen, the fragments are then inserted into the vector using DNA ligase. Is how then first these fragments with these vectors reunite with the help of the ligase enzyme, and then inserted into the into the bacterial cell for the amplification purpose. So you can easily construct your genomic library by using this technology, which is the recombinant DNA technology. Is it clear? Okay. Now, what is cDNA library and why is it important? Now, you can easily compare the cDNA library with the genomic library. A cDNA library is a collection of the cloned DNA sequences that are complementary to the messenger RNA. As I have discussed, that the cDNA library is constructed by using the reverse transcription mechanism. In reverse transcription, what will happen? RNA is converted into the DNA, and that DNA is the cDNA, which is the complementary DNA. cDNA is often used to clone eukaryotic genes in the if you want to express the eukaryotic gene in the prokaryote, you can use the cDNA. Is it clear? When scientists want to express a specific protein in a cell that does not normally express that protein, Is it clear? They will transfer the cDNA that codes for the protein to the recipient cell, like the bacterial cell. If you want to express the human insulin producing gene into the into the bacterial cell, you can easily do this by using the cDNA library. Is it clear? Or many more. I'm just giving you an example. Okay. Now, how cDNA library is constructed? That is very important. Creation of the cDNA library start with the messenger RNA. And I have discussed in detail about the protocol for the extraction of the messenger RNA from the from the cells or tissues. So must watch my lecture related to the RNA extraction. So messenger RNA carries encoded information from the DNA to ribosomes for translation into the protein. Is it clear? To create a cDNA library, these messenger RNA molecules are treated with the enzyme reverse transcriptase, and reverse transcriptase are present in most of the viruses, which is used to make the DNA copy of a messenger RNA. Is it clear? A cDNA library represents a sampling of the transcribed genes. Must keep it in your mind. cDNA consists of what? Sampling of the transcribed genes, whereas the genomic library include the untranscribed regions. Is it clear? Because cDNA is synthesized from the messenger RNA, and messenger RNA is just transcribed. Is it clear? So that messenger RNA responsible for what? For the protein. So that cDNA easily do the mechanism in any recipient cells, like in bacterial cells. Okay. Now, overview of the cDNA. First of all, you can see here the messenger RNA. This messenger RNA, by the help of the reverse transcriptase, is converted into the cDNA. Then this cDNA, along with the vectors in which the cDNA is cloned, is digested with the restriction enzyme. Then both cutted pieces of the fragments of the cDNA and the what the vectors reunite by using the ligase enzyme, and then inserted into the bacterial cell for the amplification purpose. So in this way you can easily construct your cDNA library. Now I'm going to discuss each and every step of the cDNA library in detail. So first is the isolation of messenger RNA. So you can easily extract the messenger RNA. First step is what? The extraction of the messenger RNA from the cells and tissues. Then now what will happen? It involves the isolation of the total messenger RNA from a cell type or tissue. As we are familiar that along with the messenger RNA, there is a transfer RNA and ribosomal RNA, as we are familiar that 80% of the total RNA. So ribosomal RNA and transfer RNA easily removed from the messenger RNA by using different kits. I have mentioned it by gene, and are using magnetic bead based depletion kits by chromatography too. As we are familiar that the messenger RNA are composed of strings are 50 to 250 adenylate residues by poly-A tail. This poly-A tail is absent in the ribosomal and transfer RNA. So messenger RNA can easily be separated from both ribosomal and transfer RNA because of the presence of the poly-A tail by using the oligo-DT tag. Okay. Now what will happen if we put the total messenger RNA containing messenger RNA, ribosomal RNA, transfer RNA into the column, and then we put the oligo-DT. So the thymidine of the oligo-DT form the hydrogen bond with the poly-A tail of the messenger RNA. Then what will happen? All the other RNAs like ribosomal and transfer RNA do not possess any poly-A tail, will flow through the column as a waste. Is it clear? Okay. Now in this column you just have messenger RNA. Now what will happen? The number of the desired messenger RNA can be increased by the following methods. You can use the chromatograph method. This is the chromatographic method, and you can also use spinning down messenger RNA using the gradient density gradient centrifugation. You can also get the messenger RNA by this method. Okay. Now, how can you prepare your reaction mixture for the synthesis of the cDNA? We need the reverse transcriptase enzymes, we need messenger RNA, we need the buffers, we need the DNTPs, we need the DNA polymerase, and we need the primers. Is it clear? And this involves the one-step RT-PCR. So I'm not here discussing the PCR mechanism, just I'm showing you the reaction mixture is then put into the PCR tubes, and the PCR tubes then placed into the thermocycler for the reverse transcription and the amplification of the cDNA. Is it clear? Okay. Now, synthesis of the first strand of the cDNA. Messenger RNA being single-stranded, is it clear? And consists of just the exons. So this single-stranded can not be cloned and is not a substrate for DNA ligase. Is it clear? It is first converted into DNA prior to insertion into the suitable vector. So we have to convert first messenger RNA into the DNA in order to insert into a suitable vector. So we need the, as I have discussed, that oligoDTs, reverse transcriptase, and we need the DNA polymerase, we need the primers. So what will happen when the messenger RNA, I am showing you here, you can easily see that this is the messenger RNA. This messenger RNA in the reaction mixture in the PCR is converted into what? Into the cDNA by the mechanism of reverse transcription. Is it clear? So what will happen? This cDNA and messenger RNA form the hybrid, and this hybrid is separated by using the enzyme RNase H. Is it clear? RNase H is used to give a single-stranded cDNA molecule. Okay. Now what will happen? Now this hybrid is is removed. Means the messenger RNA is removed from the cDNA by using the H enzyme, RNase H enzyme. Then now what will happen? This single-stranded DNA, cDNA is converted into the double-stranded cDNA by the help of the enzyme, which is the DNA polymerase enzyme. Now what will happen? These two strands are joined, form the loop, and this loop is cut by the S1 nucleases. Now the cDNA is now ready for cloning purposes. Is it clear? Okay. The second strands of the cDNA generation, after the formation of the first strand, you can see here the second strand is formed, and by the help of the DNA polymerase, and then these strands loop is trimmed by the S1 nucleases, and at the end double-stranded DNA is produced. So you can read the slide and note down the information. Okay. Now the incorporation of the cDNA into the vector. Now the double-stranded DNA, which has been trimmed with this S1 nuclease to obtain blunt-ended double-stranded cDNA molecule, followed by the addition of the terminal transferase to tail the cDNA with C's and C means cytosines and ligation into a vector, because the blunt end ligation is inefficient. Short restriction site linkers are first ligated to both ends. Is it clear? On both ends of this cDNA, what will happen? The restriction site linkers are first ligated on both these ends. Is it clear? Linkers are added. For what? For the insertion of the double-stranded DNA into the cloning vector. So you can see here how this cDNA is cut and then linkers added, and after the addition of the linkers it is inserted into that vector, and this vector and the DNA is reunite, joined with the help of the ligases enzymes, and then inserted into the what, into the vector, into the uh, bacteria for the clonal. cDNA are commonly cloned in the phage insertion vectors. Bacteriophages vectors possess the following advantage over the plasmid vectors. Bacteriophages are more desirable when a large number of the recombinants are required for cloning. Low abundant messenger RNAs as a recombinant phages are produced by in vitro packaging, can easily handle and store large amounts of the phage clones as compared to the bacterial clones carrying plasmids. So there is a comparison where you can easily note down the genomic DNA and cDNA library construction. Here the simple DNA is cut into the fragments. Here the messenger RNA is first converted into cDNA. Then this cDNA digested with the restriction enzyme and then inserted into the required desired vectors, and these vectors now inserted into the bacterial cell for the amplification purposes, or for, you can say that, for the cloning purposes. I hope so the lecture will be clear for you people. If you have any question related to the cDNA library or genomic library, you can ask and write in the comment box. I hope so the lecture will be clear. I have tried my best to give you the clear concept related to the genomic library and cDNA. So I will be back with the randomized library in my next lecture. Till then, Allah Hafiz.